Today, we're going to talk about an organism that's been hiding in plain sight for years, Pseudomonas monteilii. Now, this is an emerging pathogen that's often missed, frequently misidentified, and, frankly, widely misunderstood. So let's break down exactly why your clinical lab really needs to have this bug on its radar. And just so you know where all this info is coming from, this entire explainer is built from a comprehensive review we compiled using Gemini Deep Search. If you want to get into the weeds and check out the primary literature yourself, the full list of sources is right there for you in the accompanying notebook. Okay, let's just dive right in with a question. What if a routine lab result, one you probably see all the time, is actually masking a much more virulent and clinically significant pathogen? Well, that's the exact problem we're facing with Pseudomonas monteilii. So here's our game plan for today. We're going to start with its case of mistaken identity, then we'll move to what it looks like on the bench. After that, we'll cover the clinical diseases, talk treatment and susceptibility, touch on infection control, and then we'll wrap it all up with the key takeaways. All right, first up, a case of mistaken identity. This is really the core of the problem with this organism. So what exactly is Pseudomonas monteilii? Well, it's a gram-negative opportunistic pathogen. But here's the crucial part. It belongs to the Pseudomonas putida group. And its close genetic relationship to Pseudomonas putida is precisely why it's been living in its shadow, historically under-recognized, because, well, our older methods just couldn't tell them apart. And this slide, it just shows the problem perfectly. For years, conventional systems like the Vitek2 would spit out a result saying Pseudomonas putida. It really wasn't until the widespread adoption of MALDI-TOF MS that we could reliably see the true culprit, Pseudomonas monteilii. Getting the name right, that's the critical first step to understanding its real clinical impact. So let's head over to the bench and see how this all plays out. Section 2, identification, from the culture plate all the way to the sequencer. On the bench, it looks like a classic non-fermenter. It's a gram-negative rod, it's motile, and it's an obligate aerobe. On McConkey agar, you're going to see those pale, non-lactose fermenting colonies. On blood agar, it's non-hemolytic. But here is a key differentiator. Pigment production. It produces that fluorescent yellow-green pigment, pyoverdine. But, and this is critical, it does not produce pyocyanin. That blue-green pigment that just screams Pseudomonas aeruginosa? And that brings us to a really simple but powerful differential test. Just take a look at this table. The key way to tell it apart from its more famous cousin, Pseudomonas aeruginosa, is temperature. Pseudomonas monteilii fails to grow at 41 or 42 degrees Celsius. This is a reliable, low-tech way to distinguish it from the thermotolerant Pseudomonas aeruginosa. A super simple test with huge implications. But for a definitive ID, we've got to go molecular. While 16S rRNA sequencing is great for figuring out the genus, it just lacks the resolution to tell the species apart within the Pseudomonas putida group. So what that means is, for an accurate species-level ID, you have to sequence a more discriminatory housekeeping gene, something like RPOD. And of course, whole genome sequencing is still the ultimate gold standard for reference and research. Okay, so we know how to find it. Now, why does it matter so much? Section 3, the clinical disease spectrum. This is where we see it go from just another environmental organism to a truly invasive pathogen. And the range of diseases it can cause is genuinely alarming. This is not just a contaminant, folks. Published case literature has documented Pseudomonas monteilii causing fatal meningoencephalitis even in immunocompetent people. It's a known cause of device-associated sepsis and bacteremia, and it can trigger really severe exacerbations in patients with bronchiectasis. The fact that it's been isolated from sites like the placenta, bile, and fluid just further proves its invasive potential. Yeah, this quote from the source material really drives the point home. The ability to cause severe disease even in healthy hosts is what elevates this thing from a simple opportunist to a pathogen of pretty significant concern. Any isolation from a sterile site has to be taken very, very seriously. So how do we treat it? Let's move on to Section 4, treatment and susceptibility. This is all about understanding its resistome and figuring out a smart therapeutic strategy. So Pseudomonas monteilii shares that pretty formidable intrinsic resistance of its relatives. We're talking about low outer membrane permeability thanks to porins like OPER-D, powerful multi-drug efflux pumps like MEX-ABORM that basically just kick antibiotics out, and an inducible chromosomal AMP-C beta lactamase. This baseline resistance makes treatment inherently challenging from the get-go. Now listen up.
because this is absolutely critical for the lab. As of 2024, EUCAS-CAST has no species-specific breakpoints for Pseudomonas monteilii. I'll say that again. You must not use the Pseudomonas aeruginosa breakpoints. The correct procedure is to use the breakpoints for the broader category of Pseudomonas spp. Getting this right is completely essential for correct patient therapy.
So, what does treatment actually look like? Well, as always, it has to be guided by susceptibility results. As you can see here, first-line options often include agents like piperacillin tazobactam or cefepime. For more resistant isolates or really severe infections, we escalate to carbapenems like meropenem. Oh, and I want to draw your attention to the notes on extended infusions. That's a key strategy to optimize pharmacodynamics and overcome that borderline susceptibility. Now, in the pediatric population, the story is often about indwelling devices. These infections are very, very frequently linked to central venous catheters. This makes CVC removal a critical, absolutely non-negotiable part of management. The typical antibiotic course is around 14 days, with the drug choice, of course, dictated by susceptibility testing. Okay, let's shift gears and talk prevention. Section 5, infection prevention and control. And the key here is targeting its environmental reservoir.
You know, standard precautions like hand hygiene are fundamental, of course. But because Pseudomonas monteilii is a waterborne pathogen, we have to go further. Effective IPC really requires a comprehensive water management program. And what this means is we have to focus on the meticulous cleaning and disinfection of sinks, drains, and plumbing systems to minimize the risk of splashing and aerosolization, which is how it can get transmitted in the healthcare environment. All right, let's bring it all together in our final section, key takeaways. This is what every single microbiologist needs to remember about Pseudomonas monteilii. Here are the 4 big points. 1. You cannot rely on conventional biochemical ID. Accurate identification requires modern methods like MALDI-TOF MS. 2. This is a virulent pathogen, not some benign contaminant. Treat its isolation from sterile sites with respect. 3. Susceptibility is totally unpredictable. Antimicrobial susceptibility testing is mandatory for every single clinically significant isolate. And 4. Prevention isn't just about hand washing. It must target the water-based environmental reservoirs where this organism loves to thrive. Ultimately, it all comes back to us in the lab. Accurate identification is the first and most critical step in patient safety. It changes how a result is perceived, how a patient is treated, and how we control its spread. So we'll leave you with a question for the comments. What's your experience with non-aeruginosa pseudomonas at your hospital? Hey, if you found this explainer valuable, please subscribe for more deep dives into clinically relevant microbiology. And definitely share your insights, your experiences, and even your local protocols in the comments below. Let's keep this important conversation going. Thanks so much for watching.